Hey, you want to do an introduction? Yes.
Alright, thanks everybody for joining us today. I have the pleasure of introducing Dr. Jason George. So he joined us from Texas A&M University, where he is an assistant professor in biomedical engineering. He received his BA in mathematics and BS in biomedical engineering. Well, he earned his PhD in bioengineering from Rice University in 2019 and his MD from Baylor College of Medicine in 2020. He completed his postdoc studies at Rice University and MD Anderson in 2021 before joining the faculty at Texas A&M in 2022. I had the privilege of serving as a co-leader for the Math Onco subgroup for SMB with Jason back in 2022 to 2023, I believe. And he is still the subgroup leader right now. What else? His postdoc Pujan was also here for the IMO workshop, if you all remember. He was in our group last year, so that was fun.
Actually had 3 people here for the workshop.
Yeah. What else? So his research group focuses on developing and applying mathematical and statistical models to explain and predict the behavior of complex biological systems. And today he'll be discussing his work on biophysical and stochastic modeling of the tumor-immune interaction for optimizing T-cell immunotherapies. So please join me in welcoming Jason.
Thank you, Renee, for the wonderful introduction. And I had a blast as co-chair with you. And really, I think that was my first foray into mathematical oncology, was this SMB group. Since then, I mean, our group and I think Texas A&M and our department has really been impressed with a lot of the initiatives that are coming out directly from Moffitt here in this program. Yeah, all of my trainees had a blast at this workshop, so I am preaching it out to everybody who's possibly interested. I think it's fantastic. And I also want to just take a moment to say how I think special, which is something I think everybody here appreciates, but it really shouldn't go without saying that the culture of having an integrated group where mathematicians and modelers are really discussing actively with clinicians who are interested and engaged, and the interest and engagement is mutual, right? Is really what I think our field needs to be driven forward. Without that, you have sort of people working in silos. And so I've only been here, I mean, physically for 3 or so hours, but already it's immediately clear that's a true gift of this department. And so I'm just really astonished by this and think it's very special. So and also by way of introduction, I have my MD there. So when I was, graduating from undergraduate, I sort of was indecisive. I knew I wanted to help people, but I also loved math. And this sort of gave me a way to do both simultaneously. And, going through the clinic, I appreciated, problems that for me, the most interesting ones were cancer and how to manage that from a clinical perspective. But my true love was really in modeling and trying to provide some foundational insight into an underlying disease process with the hope of optimizing therapy. So I made the giant leap to, sort of circumvent all of the clinical training after medical school. I didn't go to residency and then went straight into research. But given this sort of, dual training, I've really seen that the languages are different, right? The priorities are often different. So it's non-trivial to align all of that. Now, this talk in particular, our group is a relatively young group, is interested in a number of things ranging from theoretical sort of model-driven design to understand important biophysical or biological problems to more inferential statistical or data-driven problems. And so today I'm going to be talking about, one case that really the story started at the very end of my PhD and has continued through the postdoc and now into my early independent career on biophysical modeling of how we think about the tumor-immune interaction, and in particular, how we think about T cells interacting with tumors via targeting of tumor-associated antigens. Okay, so let's see if I can. Okay, all right. So we'll start out, the outline here basically is a little bit of, motivation and background, in particular motivating what is T-cell immunotherapy, why is this important, and sort of what are the variety of T-cell immunotherapies. And then I've structured the remainder of the talk into 2 pieces. The first one is basically on how do we model T-cell recognition of cancer via these antigens? How do we think of that from a kind of first principles approach? And then how do we sort of develop it into something that could be used as a tool, perhaps a prognostic tool for a clinician that's worried about cancer progression. And then in the second part of the talk, I'll discuss this kind of population dynamical modeling of the tumor-immune interaction that leads to what I like to call sort of this cat and mouse chase between an evolving cancer that presumably tries to evade detection via, either clinically given drugs or an innate immune system. And then the adaptive immune system, which can really select for T cells that are specific to a variant that may emerge. Okay, so I skipped the slide that says cancer is a problem, because I think everybody here understands and very well appreciates this. If I may borrow from Hanahan and Bob Weinberg, this sort of now classic picture of the hallmarks of cancer. There are a variety of mechanisms that tumors utilize to avoid either recognition or control or detection, and then they gain sort of a proliferative advantage. For the sake of this talk, the one that I really want to focus on exclusively here is this hallmark wherein tumor cells, by definition, must necessarily avoid immune targeting if they're to be successful. So where this all starts, and in particular, the talk is going to be referring to the immune system interacting, in the adaptive context with T cells, and in particular with cytotoxic T cells. So these T cells, basically are part of the adaptive immune system, which the purpose of this immune system is to essentially recognize threats that have never been detected before, right? That have a unique signature, control those threats, eliminate, the virus or whatever the stranger signal is, and then develop a memory so that, were these threats to reemerge at some point in the future, they could be capably cleared with this adaptive system. And so key to this, adaptive system is in the T cells, which operate primarily by this TCR, T cell receptor, or TCR for short. And so you're going to see this probably repeated as kind of this bubbly cartoon in my talk. And so this is really key to their recognition. So T cells have a TCR, a unique TCR, and there are many TCRs uniquely in an individual. And I think by some order of estimate, the number of unique clones in one given, person is of order 10 to the 8th to 10 to the 10th, depending on how diverse their repertoire is. And so each of these have a unique TCR. And basically what happens is that this TCR, this T cell, will go around in circulation surveying cells by interrogating or interacting with an antigen that's presented on this major histocompatibility or MHC class 1 molecule. And so the idea is that these antigens are signals from inside the cell. They're digested peptide fragments on a CD8 T cell that we're interested in. They're comprised of 9 to 11 amino acid fragments. And the idea is that the T cell needs to make a decision. Basically, does this look like a normal protein fragment from the cell? If it does, the T cell wants to leave it alone. If it looks foreign, perhaps it's an antigen from a virus that's now infected the cell, the T cell wants to eliminate it. And the T cell sends a killing signal to this presumably infected cell. And this is what's responsible for imparting durable immune safety against intracellular pathogens like viruses and intracellular bacteria. What makes this interesting from a modeling perspective is the diversity. So as I said, and this number is conservative. So 10 to the 8th to 10 to the 10 in very diverse settings, unique clones in an individual. The upper theoretic limit on TCR diversity is quite staggering. It's something of the order 10 to the 17th. So even one individual only has, a very, very small fraction of all possible allowable T cells. And presumably, on a population level, many individuals can cover that space. The other thing is that these antigens are ordered sequences of amino acids. So the total upper limit on diversity there is of the order 20 to the 10th or 10 to the 13th. So this is exciting from a modeler's perspective because there's no hope that somebody in a sort of experimental context can reliably characterize this problem completely, right? I mean, it's just the order of magnitude won't work. And so we wanted to sort of understand this at a basic level. So as I mentioned, this is what sort of imparts immunity against foreign antigens. So this is a picture, an electron micrograph of a T cell attacking, killing, recognizing a presumably infected cell. by basically interrogating and finding an antigen that's a viral antigen. And as it turns out, the same exact procedure we now can appreciate can operate to confer durable immunity against tumors. So, although a tumor is a self-cell, the mutational process of tumors can result in self-signatures that are normally tolerated to a T-cell recognition event to become recognized in the form of what we refer to as tumor-associated antigens. And so for conceptual understanding in this talk, we really sort of think of these as two different categories. One where the mutational process of cancer imparts some sort of point mutation to a protein that then gets expressed and then finally manifests as an altered self-peptide. And these are new peptides that the immune system has never seen before. So they're called neoantigens or tumor neoantigens. And it looks 90% alike to the self-signature, right? But it's just mutated in one spot. So presumably that mutation may possibly confer a recognition signal to T cells. And then the other kind is more like a foreign or random sequence wherein the host, again, through the process of cancer progression, may overexpress or mislocalize normal self-peptides that would otherwise not be displayed. So they're sort of, although they're they're not normally seen by the immune system. The immune system has no tolerance mechanisms to them. And so they really look like something that the immune system hasn't been tolerized against, right? So we effectively see these as sort of foreign-driven peptide signatures. And so these are sort of our two methods of displaying these. And of course, this is very different than something in this list. This is very similar to something on this list. And the question is, how do tumors, effectively or possibly ineffectively recognize each of these cases? Okay, so, and before I continue, I just want to mention that in this talk, when I say T-cell immunotherapies, I'm specifically thinking about therapies that enhance or augment this anti-tumor T-cell response. So basically, if we can increase the level of killing that the T-cell repertoire, all of the T-cells in a host or patient can achieve, these are the strategies. And there are a number of strategies. Checkpoint blockade is not on this list, although perhaps that's one of the more famous ones. We have tumor antigen vaccine therapies, which essentially operate to basically inject a set of tumor-associated antigens into the host with the hope that this stimulates an immune response from pre-existently specific T cells, right? You have the T cells there, they just haven't been stimulated, but they can recognize. So this works in a sort of very similar fashion to a traditional antigen vaccine. And then a very kind of science fiction, very fancy version of therapy is the chimeric antigen receptor CAR T therapy, right? So this is a genetically engineered T cell with a known specificity against a target on the tumor cell. And really the hope is that this hybrid receptor, which is more of an antibody-like receptor, can bind specifically to the cancer and leave everything else in the body alone. Oftentimes, autoimmunity and off-target effects are an issue here. This is a highly specific version of T-cell immunotherapy where you're going after one target. And then perhaps, in my opinion, one of the more interesting therapies is this idea of hematopoietic stem cell therapy. So really this is a treatment for, a lot of hematological disorders where you chemoablate the bone marrow and you need to you replenish the bone marrow with a functioning immune system. And you do that with a donor-derived allogeneic graft, right? And so as it turns out, this kind of represents, inserting a completely new T-cell repertoire into the patient. And the idea is that perhaps where the original T-cell repertoire failed at recognizing cancer, we may in fact have a chance that the new repertoire can recognize that tumor. And in fact, that's known that oftentimes, providing HSCT gives durable remission outcomes, most notably in acute myeloid leukemia. So this is sort of, this spans the gambit of some interesting T-cell strategies. Okay, so let me now move on to sort of the structural modeling part of this talk, specifically, referring to the T-cell interaction with peptide. So maybe one more background slide. So one question is, how do T cells learn what to target and what not to target? It actually turns out that the answer is more complicated than what's presented here. There are two mechanisms. One is a central tolerance mechanism, which is this mechanism. So in central tolerance, developing T cells migrate from the bone marrow to the thymus, and they undergo a selection procedure, a double selection procedure. So in the first part of this, and this occurs in the outer cortex of the thymus, T cells are migrating and they try to see if their randomly generated T cell receptors can successfully recognize or interface with an MHC present in the individual. And if they can, this sends a survival signal to the T cell, and it can continue on. If they can't, if there's faulty interaction with that MHC, the T cells die by neglect. Now, T cells that make it out of this outer cortex then sort of make their way through this inner thymic medulla, where medullary thymic epithelial cells are essentially displaying a ton of different proteins that are expressed in the periphery, right? So from the gut compartment, from the sort of all of the places where you would expect potentially there's an immunological reaction against a foreign entity, lung associated, right? I mean, breathing in, and then epithelial associated peptides. These are all being displayed here. And the idea is that these are self signatures that are required to live. So if the T cell can bind that and it becomes activated, that's an autoreactive T cell. Those T cells are deleted. And so at the end of this process, we have T cells that can recognize effectively or interface with MHC, but on a core group of antigens that are important for living, they're not going to recognize those. And then those T cells then make their way out. So the other part of this is that there's a peripheral tolerance mechanism that operates in addition to this in the periphery. It's a more complicated and sort of more dynamical problem. But at least focusing on this sort of core central tolerance mechanism, our first natural question was, given this process, how does that affect the ability to recognize tumors, right? And in particular, as I already mentioned, tumors might be neoantigens. So presumably they could have just been point mutated versions of something that T cells were trained to avoid recognizing in the first place. So our approach to answer this was in developing a probabilistic model of T cell selection and antigen recognition. Okay, so this was sort of again toward the end of my. Yes.
So if you just go back one slide.
Yep.
So that outer cortex, inner medulla is the physical geometry of the thymus.
Yes, that's correct.
And so that trajectory that you're sort of, is the implication is T cell goes in, passes through the outer cortex into the inner medulla.
Right.
So it's a physical.
Yes,
Right.
yes, that's precisely right. So geometrically, I mean, T cells are basically in training migrating through this sort of two compartment physical barrier, if you will.
Yeah.
And the only way they're going to successfully be pruned or selected is if they can make it through to the center after both of these steps, and they occur subsequently, one after the other. Yeah. Yeah, absolutely. And actually, just to remark, this will be relevant in a minute, but the survival, people have done studies on how likely is it that a typical T cell emerges, and I think the estimates vary widely, but here the survival probability is something like 5 to 10% of all T cells can actually interface successfully. This number is highly variable from estimates from 20 to 70%, but give or take, half the T cells can make it out of this process as well. And so at the end of the day, on the order of something between 2 to 5% ballpark, all of the T cells you produce actually make it to a T cell that can be used for fighting infection or tumors. Yeah.
It sounds quite wasteful.
It does, yes. Yeah. I mean, wasteful in the sense that this process is random. So I won't go into those details, but there's sort of a recombination event that is generated to try to create diversity. There's a lot of interesting work on characterizing, given a sort of primary sequence for the T cell receptor, what was the corresponding probability that was generated? And that work is out of E. Cole-Normal Superior and MIT. And there they found actually that some of these sequences were perhaps miraculously biased to anyway, survive thymic selection. So the actual VDJ recombination favors the survival, right? Which in the context of a 5% survival rate is pretty profound. But yeah, that's an interesting and important question. It is an inefficient, if you will, system. Okay, so our modeling approach, and we started out, building on what others had sort of, foundationally shown. Notably, Arup Chakraborty's group out of MIT, was we could take this T cell receptor and we wanted to characterize how does it interface abstractly with a peptide bound on MHC. And there are basically, two parts of this. There's an interaction, between the T cell and the MHC, directly. And there's an interaction with the T cell and the peptide that's loaded on the MHC. Now, T cells are restricted to a given MHC once they come out of the thymus, so they're only recognizing things, right? It's rare for a T cell to interface with MHC. So if we fix the MHC, as it turns out, this part of the T cell receptor is not diverse. It's not part of the random recombination event. So it doesn't vary as a function of, T cell from one T cell to the next. The part in this recombination event that does vary is the CDR3 region, the variable region, which is directly in contact with the peptide on the MHC. And so in this language, we sort of break up the total interaction into a constant contribution to energy or interaction and then a variable contribution. And in doing that, we can ask natural questions about T cell behavior. For example, activation would occur if that interaction is above some lower threshold or critical, cutoff threshold. If we say that, then we can say, for example, that in order to survive thymic selection, we need to have that the energy or interaction of the T cell represented by an X here with self-peptide does not exceed this lower threshold for all self-peptide, right? And similarly, if we can get past thymic selection to recognize a foreign antigen would have to interact with this T cell above that very same activation threshold. So the interesting part of this is the sort of the constitutive assumption underlying this more interesting variable term, right? And so we tried a lot of things out. We were not successful with several, more simple models. So I'm going to show you the one that ended up sort of being the most realistic one. So the T cell, let's start with a peptide. So peptide in the MHC group are going to be represented by sequences of amino acids, just as they appear. In the early version of this model, the T cell itself is represented by a matrix of random energies. And you can think of that as drawn from a uniform 0 to 1. You can do it with a Gaussian distribution. Actually, the results look the same, with the idea that the interactions between the T cell binding groove and an amino acid are site dependent. So, for example, a valine at position 2 may not interact necessarily with the same contribution as a valine at position 7. So this IJ sort of entry in the T cell matrix represents the interaction between a particular amino acid were it to be located on the peptide at J at a given position I. And this allows us to have this kind of position-dependent interaction. Let me give an example. I think this is a little easier to see. So a T cell is generated like on a repertoire level, right? We want to simulate an entire human repertoire. Those T cells are represented by their matrices of random energies, and the peptides that we select are going to be represented by their sequence. So whenever we interrogate the total energy of interaction, what we need to do is we need to find row by row where those amino acids are in the T cell matrix. And then we take those position-specific interactions that vary as a function of the peptide amino acid, and we just take their sum. That's always going to be a sum of entries, going down the row. So the first thing we can do with this is we can say, if we simulate this process over a large number of T cells, if we're trying to, in silico derive thymic selection, what is the lower activation cutoff for T cells in order for them to recapitulate normal negative thymic selection probabilities? And in particular, we want to do this with, estimates are about 10 to the 4th self-peptides in the thymus, which isn't that much, by the way, right? I mean, that's a relatively small number of self-peptides. But per MHC, that's the typical order of magnitude. And so if we proceed with evaluating this over all 10 to the 4th, we can plot out the probability that the T cell is deleted as a function of how much energy needs to be given to activate. And so we sort of use this to read off a 50% selection cutoff. Now, for the sake of time, I mean, the advantage of this toy or foundational model is it's amenable to direct analytic calculation, which is wonderful. But we can also do large scale simulations. And I won't go into the precise mathematical details here on that assumption. I mean, really we're taking a set of 10 to the 4th independent events and estimating it based on the success probability for each of those. So it's really an extreme value distribution that we're sampling from. So this basically converges to a Gumbel distribution. But the analytics are also, something that we are really happy we can do here. So just like yes, question.
Do you include any positive selection threshold? So like anything that is too strong binding will be excluded or no?
So anything that's too strong binding is excluded. No, we don't. It is in fact known that T cells can die by activation-induced cell death. So in fact, negative selection here is only going to give you an upper cutoff on the lower recognition threshold. In this particular step, anything above that will be deleted. In recognition, though, one refinement might be saying, okay, there is a range above which anything that recognizes too highly is deleted. But those extreme values would be correlated with, globally T cells that are anyway going to be highly, activated in the self-peptide step. And so presumably they exist, but they're a relatively small contribution. Yeah, that's a great question.
I have a question.
Yes,
as well.
yeah.
For the matrix you were talking about,
Yes.
do you include, well, you said you just did like a uniform distribution. I'm wondering, because of the biochemistry of different amino acids, I'm wondering if some of them are correlated.
Yes.
What would, like,
You can add, right, precisely. So I think the question is, basically this matrix is populated by IID draws.
Yeah.
No interactions between neighbors, right? No interaction across position. You could build that in if you had an idea of how correlation could operate. We actually took a different approach, as I'll sort of refine in a moment, which was, let's replace that abstract matrix directly with amino acids that represent the CDR3 sequence. And then from there, we have basically non-adjacent spatial correlations. Yeah. So that's where we'll head there in just a second. Yeah. Great questions. Okay, so just like we can talk about, selection, we can talk about recognition. So admittedly, these are busy plots, but I'll direct your sort of attention to, on the left, we have the same plot as a function of, activation threshold, where we simulate these T cells to undergo recognition conditioned on surviving thymic selection. So the regimes of interest are given in these sort of red crosshairs. This is where thymic selection is physiologic. We have, again, analytic approximations in sort of the solid lines. But what we notice is two things. In the region of interest are simulations with very large numbers of self-peptides that are reflective of thymic selection converge to our approximation. These are approximations in both cases. And if you'll notice, the actual recognition levels for an individual T cell conditioned on surviving thymic selection between a foreign peptide and a neoantigen are not that different at all. So these graphs are actually, the ranges are comparable. This surprised us because we thought that a foreign antigen should be recognized much more, very, very differently than something that was trained nearly to be avoided in thymic selection. These neoantigens are just 1 point mutant differences. And we find, in fact, that at least if we believe the thymic selection that's operant in our in silico setup, that number is about 74% or so of the way to maximal recognition of a completely random, presumably viral epitope. So this was surprising to us, and it sort of suggested, maybe perhaps counterintuitively, that going after tumor neoantigens is a really good strategy, because the immune system, you could imagine the likelihood of recognizing something is a function of distance away from self should clearly be 0 when you're identical, right? Up to some tolerance. And then it should, kind of intuitively, monotonically increase until you're very far away immunogenically from self, right? And so this picture is confirmed in our experiments. We've done this and sort of done it in variable numbers of mutations, but the jump from 0 difference to 1 difference is huge. So it's quite interesting that the immune system, when sort of trained, can do this. Okay, and now comes, yes.
Sorry, I have a quick question.
Of course.
So this is a really cool result, very, very surprising indeed. I'm just wondering because, okay, when we have proteins, they consist of the amino acids, but they are
Yes.
also special. They are also organized, right? Like when you have a sequence, some of the sequences may not necessarily be actually valid. Some of the proteins might be misfolded, or some of them might have
So peptides, 20 amino acids, but they don't actually even assume the 3D structure most of the time. So it's actually real kind of like going and looking at all day.
Yeah, so
Especially if you have some charged amino acid,
Mhm.
just like a brand new coil. So it like really goes and looks at it. Really based on primary sequence rather than secondary structure.
Yeah, these are digested. That's a good point.
Okay.
That it's really, I mean, conditional on being put into an MHC binding groove.
Mhm.
They're kind of stretched out sequences that are anchored usually in two
Yeah.
places at the beginning and end. And it's just the digital outread of,
Gotcha.
a 10 amino acid string.
gotcha, so there is no things like, oh, we might be having misfolded protein here that can be.
Not in this context, yeah.
Gotcha.
In
Thank
fact,
Yeah.
very short peptides, yeah. In this case, 9 to 11 amino acid peptides, yeah.
Thank you.
Absolutely. Okay, but as was previously pointed out, I mean, there are simplifications here and we wanted additional refinement. So many simplifications. The ones that mattered were no correlations, right? These were random energy models. They were IID. There is spatial correlation. There should be correlation amongst similar families of amino acids, right? All of these things presumably are sort of important in the real world. And the other thing was too, that the T cell was conveniently described as an abstract binding groove, right? But we have real T cell sequences. So to refine this, and we also know that it's not as if the T cell interacts with peptide one and only one position. There's actually cross dependence in the interactions. So to generalize this model, we sort of next, re-represented T cells as real amino acid strings. And then the natural question in our minds was basically, how do we assign non-adjacent interactions to that spatial representation of a T cell? We have primary sequence information, but we don't have the secondary or tertiary sequence information. So, we've been very sort of impressed by looking at crystal structures of known T cell peptide interactions for those that, elicit a response. These are important T cell peptide pairs. And from that, we can basically ask what the pairwise distances in 3 dimensions of all of the amino acids on the T cell variable region and the peptide. And the output of that would be a contact map, right? Which is a map where entries are between 0 and 1. It's some, decreasing function of the distance, right? The further away you are, the less contact you make. And immediately we see that this contact map is non-diagonal, right? It's very blocky. So actually the first, all of the models, that I'm aware of up until this point, including our own, assumed a strictly diagonal mapping in sort of our, interactions, and it's just not the case. And this is one very representative example. So using this contact map was sort of one way to do this, and then we would train an energy model, which I'll describe in a moment. But when we do this, we can repeat thymic selection, we can repeat recognition, and basically what we see is essentially subtleties in the rate at which things are recognized or selected for or against is a function of subtle differences in a contact map. So these contact maps I'm showing are family of contact maps. This particular one shares a common antigen, but the T cell is different. And then similarly, these share a common T cell, but the antigen is different. And you can see slight variations. As you move from different functional groups, the variations become pretty profound. But within a family, there's subtle variations. So we were happy that this at least accounts for the physical topology that occurs in an MHC T cell interaction. And we have now an ability to go after directly imputation of T cell sequences that we have via primary sequence, which is what a lot of the data has. I do want to mention this. So this is relatively new work, but one thing that we do want to do is characterize the energy or binding energy variance. So it turns out that the variance in the binding energy is what matters in recognition, because you're really pooling over an ensemble of interaction events. All you need is one. So what is the maximal, right? And the wider your distribution, the more successful you will be at recognizing. And so, we were able to, solve analytically for the second moment and then the variance of these as a function of an arbitrary contact map. And here I'm just plotting basically as a function of the number of contacts, right? The more contacts, the more interactions, presumably. Each of these are all human MHC class 1 systems. And what the variance does, how does it scale as a function of number of contacts? And here are the idealized calculations for motifs that would be sort of a vertical motif in the contact map. Here's our typical assumption of diagonal, binding motifs. And then this is more of a box motif. So already we can sort of throw out diagonal contact maps as being useful from this exercise, or we think that that's probably less realistic. A lot of them seem to be boxy, but you'll notice there's a substantial diversity in the variance estimated from different functional clusters, which may represent, for example, interactions amongst T cells, and in particular, MART1 is a tumor neoantigen. So tumor neoantigens and viruses and which T cell peptide MHC they're restricted to all matters in the variance calculation through the contact map. So we're currently working to formalize this a little bit and to establish it on a lot more sequences. But now I want to talk about yeah, go ahead.
So just so I can fully understand this,
Yes.
this is obviously an abstraction of a T cell interacting with a tumor cell. And you're just looking at what, if it was an idealized interaction, what that mapping would look like to trigger behavior in the T cell.
That's right. That's precisely right. Here, the ensemble is taken over, in this case, these were realized sequences of T cells that we know against a presumed ensemble of, say, peptides that could represent a viral infection.
Right.
And the question is, okay, if you have an extreme draw of that, what's the maximum energy value? And hence we're focusing on variance. Yeah. You could do this conditioning on a particular T cell peptide interaction, which would give you a real number. Then you could realize that for a collection, for example.
Because that's where I was going to go, was like, in reality, there has to be the physical interaction between
Yes.
the T cell and tumor
Yes.
population, right?
Yes.
And how often does that have to occur for the education of the T cell population? Do you know what I mean? Because that's to build up somehow in terms of what it sees, what it identifies in the tumor, right?
Yes. Yeah. So the places where what do we neglect here?
Yeah.
What we're neglecting here is we're not saying obviously these antigens, either viral or tumor, are expressed at different levels, and there's a filter on which antigens ultimately get displayed on MHC. All of that is neglected in this picture. This is conditional on antigens making it in sufficient abundance to elicit a response. But in general, that is a non-trivial and important part of the problem. Yeah.
But the point is, it's not diagonal.
The point is it not diagonal? Yes, not diagonal at all. There was another question.
Because
Yes.
I realized my question was already answered, but I was just curious to know, if you also get box diagonals in those?
Oh, like sort of.
Yeah. So
I
have-
never. Yes, actually, there is at least. Which one is it? I think it's a tumor neoantigen. I think it's. I think it may be MART1, which I'm sort of so this is one realization of that, but there is a case where you actually have kind of two boxes side by side. Now the value of being able to solve this directly is that you can then take any topology. I mean, create it, right? It could be a box of any type, and then you can calculate the variance. And I think that would serve as sort of a surrogate measure of the, all things equal, availability of that particular system to recognize something from an extreme value. Yeah. But I haven't actually looked at, that'd be interesting to look at additional patterns. Yeah. Yeah, we were surprised at this because so far, again, everything was on diagonal. And so, yeah, there's definitely refinements. A lot of these probably have other canonical topologies that aren't in fact a box. Yeah.
And then one other question, that your energy
Yes.
abstraction here,
Yes.
right? What can we intuitively interpret that as the likelihood that interaction would occur, or?
We assume that if the peptide is present and the T cell is present, then they will interact. This energy is a comment on what is the binding affinity of that interaction. And the binding affinity is going to trigger a sort of a downstream effect for the T cell to make the decision to activate. So we're really physically thinking of this as like a KD, an affinity, right? And if we exceed that affinity, we at least have a T cell that's capable of firing against that threat. Yeah. There's been a debate, actually a long debate on whether or not affinity versus more kinetic, like off rate, is more important. I think the answer is everything is important. But we don't have the data for off rate. It's so much harder to measure. So affinity is what we're guiding just based on the availability of data for clinical application. Yeah.
Do all the positions have the same value, like out of this 10 amino acids, like are they kind of same important?
Well, yeah, so through the contact map, for example, you would say not necessarily. So if a T cell is recognizing a peptide, this position on the T cell only sees 1 point, I don't know if you can see that, but it's like 1.1 weighting of the total positions.
It's
Whereas-
very, very faint.
Yeah, I see it.
It's only now that you pointed at it.
Oh, you
Yeah.
can see it. Yeah, you can see it. It's very faint, maybe at this angle. I don't know.
Oh, yeah.
But it's variable as a function of that contact. So you can imagine that, there are several corollaries. The first being not all T cell positions are equal. And the second is depending on how much this specific peptide position is seen by T cell, you may have an easier or harder time recognizing an antigen that's mutated. Yeah. So it is non-uniform. Good questions. Any other source ideas? Okay, but our ultimate goal, in starting all of this out, was to generate something that could be used in clinic, right? You want a tool to recognize, what is a tumor neoantigen and whether or not it can be recognized by a real T cell. So we started this off, a few years ago, and this was all done in the mouse system because at the time we had more data there. And really what we wanted to do is we wanted to say, given a crystal structure that we were confident represented T cell peptide binding, together with a list of sequences, T cell and peptide pairs, that are known strong and weak binders, right? Those that are recognition versus non-recognition events. How can we train an energy matrix? This energy matrix represents how favorable an interaction between one amino acid and the other are, such that it maximizes the affinity or energy of interaction between known, previously confirmed strong and weak binders. And if we can, and that's the optimization objective, is to train this matrix. If we train this matrix, then the hope would be we can take what the trained matrix using either this crystal structure or another one that's more suitable, and then make a prediction on unknown T cell and in general unknown peptide pairs, right? If you were handed, in other words, in a clinic, a list of putative tumor neoantigen targets, right? This is the universe of all possible antigens, which is on the order of maybe 1,000, depending on it's a large number. And then the T cell repertoire, which, okay, if you're doing, adoptive T cell therapy, you may be able to choose, several thousand. If you're injecting a new immune system, like in hematopoietic stem cell transplant, you're going to have on the order of 10 to the 6th T cells that are now entering. So we really wanted to do this in a computationally efficient manner to guide, clinical decision making. So we did this in a few years back in the mouse system in class 2, and it worked pretty well. We completely failed on humans. It didn't work. It failed in part because the diversity to which sequences existed structurally in the human was, for whatever reason, so much more variable. So we had to kind of revamp this. In our decision to revamp this was based on the observation that as we took known crystal structures that were published, they looked very distinct. And basically our goal now, or our sort of optimization, was just to add in another step, which was, okay, fine, we don't have one crystal structure, but you have a universe of crystal structures that's represented, and let's train this energy matrix sort of in a crystal structure specific way as a function of the pair. And we can do that in the training step, and we can do that in the prediction step as well. And that sounds, I don't know why it took as long as it did, because it's not very much of a leap from where we were, but it took us a long time to sort of realize this and do it in sort of an unbiased way, because when you're given an unknown sequence, it's sometimes hard to decide which is the best crystal structure to use. If you start threading all of your sequences through 20 or 30 crystal structures, you're going to get false positives, right? Because you're just repeating independent experiments. So with this revamped context, the results that I'm going to show for the remainder of this project are sort of given. So the first thing was this structural diversity. What I'm showing here is hierarchical clustering on structural similarity. This is given by pairwise interactions between the crystal structure positions for two different crystal structures. And it's clustered on the similarity, and I've also in the colors here color-coded based on functional groups, meaning that these systems either share a common peptide, or they share a common T cell, or maybe they're the same, but they have a point mutant somewhere. And you can see that, in some cases, so this is 1E6. This particular case is very structurally homogeneous, and all of these, in fact, represent point mutated peptide, right? So they're very similar, but there's some subtle differences. They're nothing like the other cases, and what also makes this challenging is even within a group, this is MARP1, so this is a tumor neoantigen. The structural differences are quite apparent and very heterogeneous. Now, as a sanity check, functionally similar, peptide TCR pairs tend to cluster, which is reassuring, right? It's not like we have a large distribution of these color-coded sequences all throughout. So that was reassuring. But this really illustrates that if you want to model MARP1, you have to choose a MARP1 crystal structure. You can't use one that's 1E6 or an influenza-based structure. Okay, so this is sort of our cross-validation test, and this is a little bit busy, but what I've done is I've organized it according to functional groups. A neoantigen here. These are infectious disease associated, and the results are given as a function of binding energy. The lower the binding energy, the better the prediction. Training samples were omitted in these sort of unfilled circles in testing with filled, and the gray box and whisker plots are the distribution of null binders. So basically, what do things that we expect to be non-binders look like? And we can see we do reasonably well even when we train on about 40% of the data, across structural and functional cluster. This was sort of step one. Now we fail outright in some places, right? So in this case, we can't even, and even, let's see, there's a more embarrassing one somewhere, right? We train, oftentimes we can't even suitably predict the training set that was incorporated explicitly in optimization. But overall, you can see that we do decently well from the standpoint of recognizing confirmed strong binders. Okay. But we also wanted to test this on independent data, right? Okay, great. We can take a subset of the data that we have that we're training on. So there are several data sets that we sort of selected, similar performance on each of them. This one I'm showing is from 10x Genomics. So they published basically an affinity-driven, large-scale assay involving about 2,000 T cells with 1,700 T cells with 22 peptides. And the T cells that we're making predictions on here, this is our prediction. We have removed all of those T cells that are similar to ours, and I think it was about 2% of those T cells were shared with the training set. We're completely removing them. And a good result here, these are normalized z-scores. So a good result is that we're detecting above 1. And you can see, for the most part, we tend to do all right on most of the cases. There are several places where we just fail outright. And as it turns out, these I've sort of illustrated the cases where the structure is not a good structure. So we believe we don't have suitable structural information to make a good prediction. So we're sort of limited in our approach by structure, but conditional on having a reasonable structure, we do a relatively good job. And it's important to note that a reasonable structure need not be shared functionally. So we have some cases where we don't have a structure from that T cell or peptide, but by structural or primary sequence similarity, we're confident in making that prediction. So we were encouraged by this. Now, the last part of this, I want to talk about, okay, how are we translating this into the clinical practice? So we came with this model to a collaborator in MD Anderson who works on hematological malignancies. And they basically said, okay, great. So you can resolve several of these viral associated epitopes with tumor. And it's important for us because when we give a T cell transplant, the patients are immunosuppressed. The viral reactivation is a huge problem because when we see expanded T cells, we don't know whether or not that's a therapeutic T cell or a viral reactivating T cell. So can you, in a sense, use your model to resolve these? And so the idea was that we would take some of the data from, basically our train model with a little bit more data to sort of even out the training set from their cases, from one patient, and then make predictions on T cells that were derived from a completely different patient, where the sort of clinical lab at MD Anderson had said, we pulled this T cell out via affinity to one of the antigens. Either it's a viral antigen or it's a tumor antigen, figure it out. And so, we implemented this. This is the tumor-associated neoantigen here, the one that we really want to elicit a response to. this is an influenza, and then this is I think this is a CMV, a viral antigen. And so, okay, so when we apply this, we basically took the approach of saying, okay, in the experiment, they're basically running 3 different tests for every T cell. They're basically saying the T cell is going to be subject to all 3 antigens, and we're going to get 1 strong antigen binding, right, and 1 and 2 weak antigen binding events. And we don't observe the weak antigen binding events, but they're presumably occurring behind the strong binders. So when we went to do this, we actually repeat that experiment, in our model where we say, what's the distribution of strong binders conditional on, a MART1 peptide specific T cell? And what's the distribution of the corresponding weak binders? If I'm strong binder to MART1, I might be then I'm definitely a weak binder to flu and CMV, and vice versa. So we get both, positive distributions and negative distributions through which every T cell is basically generating 3 signatures that then we compare to these sort of positive and null distributions. The only interesting caveat that we found was MART1, as I said before, is very structurally distinct. And so when we did this, we actually see 2 kind of bimodal peaks for the strong binder distributions that just so happen to overlap with the 2 primary sequence or structural similarities in the MART1 sequence. So we needed 2 different groups for that. Nonetheless, when we do that, we can then ask sensitivity, specificity, how are we doing on ROC curves? And so this is our result on the ROC analysis. And then using our original, cutoff criteria, you can see that we tend to do reasonably well in making these predictions. So we're really encouraged by this. And, of course, the next step is then to add more antigens, right? Neoantigens, presumably, or other viral reactivation antigens to see if we can do this resolution in large scale, right? The goal being that the clinician at MD Anderson doesn't want to run an affinity-driven selection against every T cell in peptide. That's slow, but, we're a little bit faster at that. Let me check the time. Okay.
Plenty of time.
Plenty of time. Okay, all right. I try to be efficient, but I can just keep talking. So basically the other thing that we thought would be really interesting is, can this model detect basically escape events if you have a mutation on the antigen side, right? So that was kind of question 1. As we know, sometimes recognition can break if an antigen, gets a 2nd or 3rd mutation. We didn't have a lot of data for this, but there was a really nice study out there on, again, the 1E6 system, where they basically did just that. They said, we have this ALW peptide and we have a 1E6 T cell, and we're going to point mutate definitively all of the positions on the ALW peptide at every single position. So they did every single point mutation possible. And then they characterized the activity of T cells binding to those point mutants, the same T cell, for every single one of them. And they're, this is sort of their graphical output where they say anything that's in pink doesn't have very much activity, so it's reduced. And you can see that in this kind of core central region of the antigen, they're really seeing a reduction in those binding values. We can do, and it's admittedly maybe light pink here, sorry for the lack of contrast, but we can do the same in silico experiment. And we actually see, for the most part, we do very well in recapitulating what they had. Where we fail, for example, is in places like this, where we didn't have a contact in the contact map. So we have no way of discerning, differences if we're point mutating. And the next question off of this is, if you can find peptides that are point mutants that may be of concern, right? You can predict them here. If you know that they're of concern, they may pose a risk to the patient if the tumor evolves. Can you then identify corresponding mutants in the T cells that will recover the recognition? That's like saying the peptide evolved to escape. Okay, great, we have a problem. This is a big one. But can we modify the T cell in a way that would react that? Oh, and I don't have that data. That's currently what we're working on. We can literally point mutate the T cell in a very similar way to find compensatory recognition recovering mutations. So that rounds out the first part of the talk. And yes, I'll take some questions if.
I was just curious about the table. Could you say something about the unit in which it is? Like, because the 2 are different, because one is experimental, one is from the model, but
Yes.
the numbers on the right seems to be all very similar. So if you can guide us on what you look for.
Yeah. So on the left, it's an experiment. The number is the TNF alpha secretion of a T cell that's binding that. So it actually isn't even an affinity measure. It's an activity measure. So that's kind of that's a little bit different. And 100 is 100% of the activity of the non-mutated version. So in fact, you have some cases where the number is above 100, right? You can actually find antigens that overreact the T cell. On the right, our units are in Z-score, or in total, binding energy, rather. And so again, numbers that are higher are higher interactions, numbers that are lower are lower, but they're normalized to this sort of 9.7 and not 100. And so what we did was we basically said, in a similar way, we can look at the proportion of these cases that are weak binders, and we can define a cutoff on our distribution that would give us similar proportion. And what does that proportion look like if we look at the lowest, the ones that recognize the least, basically?
And the cutoff is for all the numbers or is by column?
The cutoff is by column. Yeah. So some columns, I mean, in the sort of in the middle, we have a different cutoff than on the edge. Yeah. Now you could define, so in practice, right, what we're really looking for are cases that are predicted to be low by our model, very low, and agree with cases that are predicted to be very low in their model. We don't necessarily, in looking for cases of concern, look for all of the possible ones in, the sort of intermediate case. We're really looking for cases where we have a very low score that matches to 0, for example. And that's where we're focusing on sort of the compensatory mutations on T cells. Yeah, but that's a good question. Yeah, thank you. Okay, so for in the interest of time, let me move on to this other story, which is basically on a population level modeling of the tumor-immune interaction. So we have this kind of tumor antigen recognition story. Another thing we were really interested in early on was this idea that, again, this audience is very familiar with how frustrating it is to target cancer because it evolves, right? And it adapts to treatment and it, responds. And so, of course, this acquired and pre-existent, all of these mutation mechanisms that confer resistance and a subsequent fitness advantage is sort of well appreciated and frustrating for traditional therapies. Something that we liked was the idea that T cells can repeatedly interact with the tumor, right? And it might be that today we can't fully control the tumor, but tomorrow a T cell with a different specificity comes along and eliminates the tumor. So it's that cat and mouse chase that either ends in escape or elimination. And this is really seen sort of by, this is really, put into a famous terms with the 3 E's of immunoediting. So it's known that you can, have the tumor immune interaction come together, and either there's going to be an equilibrium escape or elimination, one of the 3. And our primary question was, how transient is that equilibrium? Can it last forever? Can it not? What are the probabilities of escape and elimination? But we know that the immune repertoire can't control cancer all the time, right? Because we all have a T cell repertoire, and yet some of us do get cancer. And of course, now it's appreciated that there are immune escape mechanisms that cancer utilizes. There's upregulation of checkpoint, right? Molecules that discourage T cells from activating. There's pro and anti-tumor immune microenvironments that can modulate this. When we were starting out, our first, investigation was in this rare but kind of catastrophic adaptive mechanism wherein the tumor downregulates MHC class 1 altogether. So now you forget it. You can't recognize, peptides that are loaded on the surface because that molecule's disappeared. So I motivate sort of this toy model with that idea. So our early population dynamical model basically assumed that we have a continuous, birth-death process where, cells grow at some per cell growth rate of R. These are cancer cells. If the immune system can recognize the tumor, the immune system should be able to capably clear tumors at some, per cell rate, killing rate D, where D is greater than R. And that happens when recognition is robust and reliable. The tumors won't, or the immune system won't be able to recognize tumors if they're small. So there is a minimal recognition size. And there's a lot of debate in sort of the literature, but usually this size is growth rate dependent. So it depends on the growth rate. And then that would be the entire story, except that we do allow this kind of rare but catastrophic escape event for tumors to downregulate MHC. And in our model, if that escape event occurs, basically there's no hope for the immune system to recognize anything on that MHC. So that's on the tumor dynamics of evasion. And modeling the T cells, basically we have 2 features. I mean, the first feature is total T cell diversity, the number of unique clones you have. And that is represented by a probability that once you reach maturity size or detection size, you get a recognition event. Now, it might be that a T cell was not present or in the right context in the beginning, but a subsequent T cell could arrive that would recognize this at any point in the future. And so we also have this kind of immune turnover parameter that's idealized as a Poisson arrival of a recognizing T cell, but really relates to immune turnover. So recognition clearance probability is T cell diversity, and then we have this rate of arrival, a Poisson rate of arrival that's T cell turnover. And we wanted to investigate, what could happen in on a stochastic population dynamical level. Actually, again, this is amenable to analytic characterization, so I'm giving you the exact basically elimination probabilities for cancer. And the background elimination is in this kind of very light pepper, dashed line here. And this so it's the elimination probability is a function of how fast the cancer can grow relative to the immune killing rate. So very fast cancers on the right, very slow on the left. And going from left to right, this is of various sort of problems in the immune system. So you have a very high turnover and diversity here. In the middle, you have high diversity of T cells, but low turnover. And then you have compromises to both T cell diversity and turnover. And so what we can see then, plotted on top of these dashed lines, in red is if we assume a neoantigen vaccine therapy here, and then a CAR T that has specificity against a target that is confirmed to be on the tumor. And so you can see that the sort of the utility, again, this is a very toy model. This is just in silico, simple, no data whatsoever, is you can really appreciate differences in the immunotherapeutic strategy and conferring predicted benefits to tumors that are either indolent and slow growing or fast growing. And it does seem to be that these effects of a decreasing T cell performance are very high. The choice of this going from left to right was not by chance here. So it's known that physiologically, there are first declines to T cell turnover with preserved T cell diversity in sort of middle age, in your 30s to 40s. And then as you get older, in your 60s and 70s, you have a hit to T cell diversity. So, one question that we had was, given that we know these physiologic age-related declines in the immune system exist, can we explain cancer incidence data just using that principle alone? So we ultimately, and we've done this for other cancers too, but we started with AML for the reason that AML is mutationally a very low burden, mutational burden cancer. And so this coevolution is probably not, one or two escape events is enough to result in, recurrence. And the other thing is it's a liquid tumor. So, three-dimensional T cell infiltration, which is a huge issue in many cases, is not something we're modeling in this sort of population level model. And so the question is, how do we do if we model this? The assumptions we make are just that the risk of tumor initiation is constant for every age group. That's the only assumption. And then we take published data on T cell declines, on turnover and diversity, fed directly into the model. And then we have one free parameter, which is the basal rate of tumor initiation across all age groups. And when we feed that and we optimize the model in that regard, with regard to that one free parameter, and we can see that we do, a reasonable job explaining AML age specific incidence. And this is assuming that a 2-year-old and an 82-year-old have the same rate of kind of stochastic tumor initiation. So we were very kind of surprised that the immune sort of explanation worked well here. And the question was, how do we compare this to what is already known? And so sort of the most famous model is the Armitage multi-hit, incidence model for cancer. And so we basically compared this to multiple.
Sorry, just 1 second. So when you make these predictions on theoretical model, how many free parameters do you have?
Yes. So the only.
Obviously you're constrained by what you know, but there's plenty of, you can probably wiggle with.
Yeah, the fitting parameter is the baseline initiation. The things that were not fixed or the things that are fixed are what is the functional form of T cell decline? Mm-hmm. So you have the ability to say that at a very low T cell turnover or diversity rate, you have a different Poisson rate or a different probability. Mm-hmm. But it's constrained by the fact that we basically choose that as fixed for all values of age based on those age declines. Yeah.
So what do you make of it? Looks like there is like a decline after 90s, and this is a common across different cancers. To past like
Yeah.
in really old age, you are getting decline in the cancer incidence.
Yeah, I mean, also keep in mind here that there is, I mean, very few data, right? So this becomes very noisy.
Yeah, but then it's kind of, it's not just like in AML, it's seen across different cancers, so it's unlikely to be just a fluke.
Specific, yeah. So I think, in our model at least, the settling out of this, right? And I don't think, so our model would not then presume that it decreased unless we had sort of selection or some other feature that's occurring there. And it might be that, I mean, people who conditional on making it to 80, your immune system is such that you're gonna make it to 120, right? So I think that there is, I mean, there's a selective, there's some selection there. But really this sort of decline in increasing incidence that we see is actually dominated by the fact that T cell repertoire diversity declines kind of bottom out at a certain age. And that's that's the driving feature that's explained here. Yeah. Good question. Yeah. We too were puzzled. I mean, you're right. Looking at other cancers that you see the same trend, although we have fewer data. So this is the best fit from a least squares perspective of the multi-hit incidence of cancer model for various hits and ranging from 3 to 6. I mean, maybe it's hard to see on the slides. And I mean, really the problem with this model is it assumes that there's a convex, right? Incidence that increases exponentially as you age. There was also another paper that came out about the same year as ours that basically said that nothing matters for cancer incidence except the immune system. So we would absolutely, say the multi-hit incidence of cancer is well established and appreciated. But I think what we really are excited about here is that this points to the importance of incorporating the adaptive immune system at least into a consideration for why a tumor initiating event may progress to something that results in disease. Okay, so before I go on, so really this is telling us physiologic declines in T cell performance contribute to cancer incidence that's increased. We have a model that we think works for AML, so we'll just freeze the model parameters. We're not changing it now that we explain it well. And the question was, could we go after cases, in the literature where people are put on chronic T cell immunosuppressive therapies for perhaps decades at a time? And the answer is, yeah, so solid organ transplant recipient patients are put on tacrolimus, right? They're T cell inhibitors. And the question was, can we appreciate the relative increased risk of AML on something like a decade horizon in those patients? And so on the left is our predictions where we froze the parameters. We're just asking how T cell declines due to this treatment operate. And on the right is actual, I mean, this is clinical data from chronic transplant recipients in terms of their increased risk of AML. And so overall, on the same order of magnitude, we get a reasonable prediction for the risk of cancer for iatrogenic, right? Or I mean, basically affected by the virtue of the fact that they have a transplant and they need to be on this therapy. So this to us was sort of reassuring because both natural declines and sort of artificially induced declines in the immune system seem to agree with the increased risk of AML that we have, or at least are predicted through our model. Now, I may start going a little faster to get through this, but for the sake of time, the simplifying thing in this model is that T cell recognition in immune evasion should be a repeated event. In other words, if you have an antigen, it could be detected. Okay, the antigen that's detected is going to be, the T cells are going to eliminate the cancer. But it might be that there's a subpopulation that grows out or a new mutation that maybe it's not an MHC mutant that destroys the recognition ability, but it could be a mutant that, reduces the expression of that antigen. And now there's a new escape event and it's a non-durable evasion event that needs to be controlled by the T cells. And so in the original model, we stopped sort of at the first step. And the goal next was to then sort of play this out and say, if it's a non-durable evasion event, how long does the cat and mouse chase go on? And, where does it stop? And so, the nice thing about this, or maybe the big innovation, this was, as I was pivoting toward postdoc, was that we could take the parameters that were relevant in the original model and we could coarse grain into a discrete time branching process model where the summary, parameters are two. One is the branching parameter, and one is the clearance probability of that threat via T cell diversity. And we could look at this from sort of a stationary equation or a generating equation that gave us the long-term probability of tumor elimination as a fixed point of this contraction mapping. And so by the contraction mapping theorem, for example, this is guaranteed to exist. And in fact, when we simulate this, these were all in silico and simulations, this agrees with our analytic escape probability. And it actually doesn't take very many generations to reach that. This can happen on the order of, say, 10 repeated cat and mouse chases, right, before either elimination or escape occurs. This is maybe a more fun picture to look at because that's a discrete version of this, but really, if we were to track tumor burden as a function of time, these are stochastic numerical simulations for some arbitrary time interval, but you can appreciate the diversity here of, again, this battle between the immune system and tumor that, in many cases can grow to very large sizes. These are, again, identical initial conditions and simulations. There's just a tremendous variety in how, equilibrium could manifest in terms of escape or elimination ultimately. And importantly, in this model, it at least assumes that long-term equilibrium does not occur. I mean, you have to either escape or lose eventually. And we would predict that timing doesn't take very long, or the number of generations, evolutionary periods is not that long. The waiting times can be, variable, of course. Okay, so then we wanted to, again, take this into the clinic or make some sense of evolutionary models that we could then apply to patient data. This was a rough rudimentary analysis because in the clinic, they only had, at least for this CLL case, some measure of tumor diversity. So the cellularity they're using is inversely correlated with how many distinct targets one might have to face in controlling the tumor. Very homogeneous tumors have a cellularity of 1 and heterogeneous have low values. And they only had these distributions where they could separate it based on cancer patients that responded versus did not respond to T cell immunotherapy, basically, giving a new immune repertoire, right? And so what we wanted to do was we basically said, okay, we have this 2-parameter model, and we have an idea in the model about how the sort of control and escape can work in the long run. So we have recognition, probabilities and elimination probabilities. And we basically wanted to calibrate our model to match in parameters where the empirical, complete response rate was in the clinic. And when we do that, we can then ask, okay, subject to starting with a distinct number of, clones that need to be controlled in this model independently, what is the probability of a response or a complete response? And of course, this is, again, simulated data where once we've sort of fitted our parameters to the clinical data, we can say, yeah, as a function of, immune heterogeneity, you're really likely to not respond if you have 10 independent threats you need to control. And so that was sort of the very rough and dirty training of that model applied rudimentarily to this data. And then the next thing we could do is we could say, okay, given that we've trained that data, can we then go back and simulate large-scale simulation and say, we just have an idea based on the distribution of original homogeneous versus heterogeneous tumors, how heterogeneous tumors tend to be. Sampling from that distribution, if we provide large-scale numeric simulations, can we recover? We're going to have some escape events. We're going to have some control events. Can we just partition those escape and control events and then look at their distributions of heterogeneity? And when we do that, where in our world, the cellularity is defined by the inverse of the total number of tumor clones, we actually do a pretty reasonable job recapitulating what's seen in their work. And again, this is in a liquid tumor where presumably coevolution is a little bit more nicely approached, where we don't have T cell infiltration issues and that sort of thing. So we were encouraged by this. As it turns out, if you repeat the same analysis where you're not dealing with T cell immunotherapy and you're just looking at, say, chemotherapy, it doesn't work. Which we would expect because chemotherapy isn't a dynamical process like repeated T cell recognition.
Can I ask, just to clarify the previous slide, when you were
Yes.
talking about initial clone number, that's on the cancer side?
That's on the cancer side, yes.
And is your assumption then the evolution doesn't occur in the cancer after that event?
Good question. The assumption is that subject to an initial number of clones in the beginning, every clone can independently give rise to an evolutionary event in an identical way. So fighting an initial population of 3 clones is in a sense linked to fighting 3 independent clones, and the
So
probability.
basically the 10 will always be recognizable by the immune cells that saw them at the beginning. Is that-
One more time.
Those clones will always be recognizable by the immune system because they're related going forward.
No.
No.
In this model, yeah, that's a great question. So in this model, the only way that a clone can survive is if it somehow downregulates something that leads to an evasion event. Now, if that clone can let's say mutate, even though it doesn't need to be a genetic mutation, if that clone can acquire mutants that give a favorable interaction, now you have new subclones that arise. But we do not account for the fact that the probability of recognizing the subclones is identical to the probability of recognizing the new clones. It's not, or the original clone, it's not coupled. Even though in a neoantigen model, you could couple that, yeah. We get into that, I'll talk about that variation where we're tracking real neoantigens at the very end, but in this model.
Clones obviously are related.
Yes,
Right?
yes,
And so like that would give birth to subclones and they'd give birth to subclones again.
yes.
The reason why I asked that is just that there's, it was very recent, we had a sort of experimental presentation earlier this week, in fact, where they were showing that when they were trying to recapitulate tumor-immune interactions in animal models,
Yes,
that if they just add excess mutation,
yes,
you don't actually get good immune response.
yes.
Whereas if you sort of have clonal diversity, you get good immune responses.
Oh, interesting,
So it's
okay.
not, it's just not about enough random mutation, but related mutation.
Yes, yeah.
So
Okay.
I'm not sure your model could talk to that, but I was just asking specifically so I understood how the clones are related.
There is, right, so not all, and similarly from the standpoint of immune recognition, if we're thinking of a clone as something that looks distinct immunogenically to the immune system, there are a variety of fitness specific effects to clones that are presumably related by a core set of neoantigens, but then one has an overexpression of a new antigen, for example, but the relative contribution to overall fitness is not, it's not an IID sort of process, right? It's very coupled to, what the sort of founder clone looked like, or the parent clone, yeah.
So is this more of a numbers game then, the way you're sort of articulating this? If you just have enough diversity at the beginning, then it gets hard for the immune system.
Exactly. The immune system, this is saying, in sort of simple terms, if the immune system has to go after n independent things, it's going to be harder for the immune system to mount that response on the order of time it needs to control a threat. Whereas if you have something, say a core neoantigen on the surface of a tumor, that, maybe the tumor is heterogeneous, but the immune system can recognize that one core neoantigen, that effectively is a very homogeneous system from the standpoint of immune recognition. Yeah. Thank you. Okay, so again, for the interest of time, I just want to briefly mention that we're now doing this in spatial. So there's some very interesting collaborations that could happen here. Our primary interest is in, and again, this is work this sort of ongoing, but can we take that idea of tumor immune interactions away from liquid tumors? And can we actually consider the ECM topology and what effect that ECM topology has in the spatial distribution of, these subclones? And correspondingly, how does that affect the T cell infiltration patterns and the subsequent T cell, cancer interaction? And so, we're doing this in an agent-based model, and we're basically really, at the heart of it, trying to say, essentially, if we look at tumors conditional on their escape, conditional on coming into the clinic, can we characterize the extent of immunoediting that would have occurred in a tumor subject to its three-dimensional microenvironment, right? And then this is typically done, most famously with these tumor-associated collagen signatures that, either look random or, circumferentially oriented or then in late stages radially oriented. So we're beginning to move in this direction to explain solid tumors. The very last thing that I want to say here, and I'll try to go a little quick through this, is there is this question that in the previous models, we just idealized what a downregulation event or an escape event would be. And in reality, those are tangible neoantigens that you can detect on the cell surface, right? And so if you take a tumor and you sequence it, I mean, if you actually do the experiment, you should be able to at least estimate the number of core neoantigens that are present on the surface, right? And that core would form the basis of what the immune system could or could not recognize. And so we sort of, adapted this model a little bit to account for a state variable that represents the core number of neoantigens on the surface. Now, we assume that all of them can be recognized identically. That's an assumption for a toy model, right? We could place a fitness landscape on that using empirical data. But foundationally, the idea is that given a time interval, either the immune system will or won't recognize that threat. If it doesn't, the tumor can escape, and what you see is a consequence of immune-specific escape. On the other hand, if the tumor can recognize in a given interval at least one tumor-associated neoantigen on the core of the tumor cells, the tumor cells have to scramble, and at least one of them has to downregulate that. And so the cancers can lose via, this is again sort of a direct consequence of immunoediting, that targeted tumor-associated antigen. And the process can repeat and so on and so forth. Now, the only thing here is that cancers are mutating. So it may also be that the amount of neoantigens that you have tomorrow subject to immunoediting can also increase because of the basal rate of tumor mutation. So we allow for basically, a new antigen arrival rate. And we were interested in a very specific question from a mathematical perspective here, which was, if we assume that the tumor is doing this passively, in other words, it has a fixed mutation rate, it has a fixed, adaptation rate, and a fixed number of new, mutants arriving for new antigens, how does that compare to the alternative hypothesis where the tumor can actually sense the amount of inflammation, destruction, immune destruction in the environment and modulate its evasion rate accordingly? This is actually known in human cells. There's sort of a linear dependence on the inflammation and the mutagenesis. There are programs that can ramp that up. And so presumably if the immune system is killing and the tumor is a rheostat, detects inflammation, perhaps the tumor can kind of adapt to that in real time. And maybe it survives to see another day today when there's aggressive targeting, but pays a price in more mutations that could manifest as tumor-associated antigens in the future. So that optimal program is what we were interested in contrasting with the passive case. I'm going to skip a lot of math for the sake of time, but the way that we think about this is through stochastic dynamic programming, where we have an optimal decision tree with a given probability of escape, elimination, or equilibrium subject to this program occurring over perhaps many generations. And again, this is the math person in me loving math for the sake of math. So you can write this out. You can, under certain circumstances, solve it in closed form. And you can get an idea for what the optimal policy would be for, escaping, right, from the tumor side. If you look at that, that policy is a function of high recognition pressure or low, and these are for various realizations of a total number of antigens, right? So there's some nice idealization for limiting in the regime of very high antigen burden. You can actually characterize, the tumor should respond dynamically, and their response should be higher if there's high recognition, and their response should be higher if they currently have a higher number of targets that are being seen by the immune system. And so this sort of scratched our mathematical itch, if you will. But the other thing, and again, for the sake of time, I'll just briefly mention that once you have this framework, you can ask relatively simple questions saying, how do these do? Optimal versus passive. Do optimal survive more? How long does it take for them to escape? And can we answer that question subject to a variety of recognition strategies that may be implemented in the clinic, right? And the answer is yes. As a sanity check, yes, they survive more often, and they're more characterized by long escape times if the tumor is to survive. So they basically live to fight another day with a higher chance of escaping. Yeah.
Sorry, would you please say what do you mean about immune recognition in the previous slide?
Yeah. So immune recognition. This parameter is the probability, again, independent probabilities that are identical for every tumor antigen, because we're not imposing a fitness landscape, that the immune system can recognize a particular antigen. And so if you have N of these antigens, you have a higher chance of recognizing at least one, basically.
Yes.
Yeah.
So in your stochastic modeling, every tumor cells and every immune systems are in contract, but there is some probability to be recognized or not.
That's correct. And this is being done at the level of core neoantigens on a minimal set of neoantigens on a tumor. If you have a heterogeneous tumor that has distinct neoantigen expression, you would need to calculate this in series, right? Yeah. Uh-oh. Let's see, can I escape? I think that was an ignore, so let's see if I can stay on, but okay, so yeah, does that answer the question?
Yeah. Like, in an immune tumor system, what I haven't found in your presentation, the space, because like the space kind of matters here.
Yes.
But every cells here are in contact, but there is some probability to be recognized or not. Essentially, if you wanna, for example, focus on the heterogeneous tumor or homogeneous one, still the space actually has an important role.
Right. For solid tumors.
For solid tumors, yeah.
Yes. Yeah, so for example, in order to do this in a melanoma, for example, where you would want some spatial idea of how these interactions can occur through infiltration, right? Which is that step that we're doing, but we haven't, I mean, we're almost done with that, but we have not attempted integrating this sort of, liquid, well-mixed model with that. Yeah, absolutely.
Yes.
Yeah. Okay, so let me see. I think I just basically want to say here that, this model sort of would predict that if you have very effective recognition, that is a very good immune system, presumably the probability that you get tumor evasion is lower, right? Because the idea that you wouldn't recognize a neoantigen that would be targetable if you have a diverse immune system is low. But conditional on escaping, like if you're unlucky enough to be one that stochastically escapes, given the high immune performance of your body, the number of targetable neoantigens that you have post-escape and therefore, ready to be intervened by on the part of the clinician is lower. So immunoediting kind of cuts both ways. I mean, you have a higher probability of not ever having to deal with the tumor to begin with. You would never know. But if you escape and you do come to the clinic and your immune system is well functioning, you're one of the unlucky few that have a harder to treat disease. And the balance of that is this can be, at least in this model, sort of reversed by having higher penalties paid for the immune evasion on the part of the tumor. So if, and again, motivate this with like melanoma, if it's very mutationally dirty, then of course, over time, you would expect more targets to emerge. So we think it's, striking a delicate balance between recognition and generation. The last point I want to make here is that what is really interesting is you can take the passive case where tumor is non-dynamic to response, and you can take the active case, the one that we've been focused on to this point. And in the passive case, the only thing that happens is that the immune system will generate stable distributions for the number of long-term neoantigens. In the active or adaptive case, we actually have this partition, like critical transition point where if immune recognition, this is a critical recognition rate. So if the immune recognition is very efficient, we expect control to where the convergence basically of the number of antigens that are targeted over time, it actually turns out that this is like an AR, like a mean reverting process. If it's below this critical rate, that becomes an unstable point. So that actually drives long-term, prior to coming into the clinic, either a really antigenically, as predicted by this model, an antigenically rich case, or one that's very depleted. And the other thing that, the other parameter, because as mathematicians were obsessed with minimizing it to key parameters or summary parameters, is this is for basically effective or ineffective immune recognition. Perhaps this is something that we could think of as an infiltrated versus an excluded tumor in a 3D tumor context. You get a lot less recognition if you can't get to it physically. But there's also this kind of microenvironmental effect where how penalizing is it for the tumor to respond? I mean, if the tumor is going to have a hard time and have a lot of mutations, then this may be speaking to anti-tumor versus pro-tumor sort of partition. So my last slide here is just to say that you can take a very kind of toy model using these critical transitions, and you can ask, okay, given that we have these two parameters, how does the baseline case, where baseline just means that we have an infiltrated tumor where the microenvironment favors recognition or elimination of that tumor, and how is that dynamical picture modulated in time if you prevent T cells from recognizing, or if you allow the immune microenvironment to favor the tumor, or perhaps both, and you get this kind of splitting behavior. So this behavior was, again, specific to adaptive therapy. So the real question at this point, and we're working with experimental collaborators to validate some of this in vitro experiments. I mean, it's still far away from an in vivo application, but can we see this kind of divergent behavior under critical transitions on our parameters in the model? And so, okay, so I think I took us up to 1:30, and I appreciate the questions. That was actually a wonderful discussion as we go. So with that, I'll thank my collaborations and support. I've been lucky to so far have recruited a very wonderful group of graduate students and postdoc. I think several of whom you all saw in the fall, and in the future I hope to send more back because they loved it. And then our scientific and clinical collaborators, and I'll acknowledge funding from the state of Texas, and we'll take any more questions at this point. Thanks for your attention. It looks like maybe, I don't know if you guys can see this, there's a chat.
Oh, yes,
But maybe I try to. Hi, Heiko. I wonder what an information criteria analysis would do, and this was at 111. I don't remember which slide I was on for this. Sorry, I didn't see the question earlier.
Hey, Jason. Sorry, Hey,
Hey,
when you showed your AML incidents and your model had this gorgeous fit, and then you did some of the other analyses of the two-hit model.
Yes.
Um,
This one here.
Yeah, that's right. Was wondering what's the number of parameters in each of those models how much information is in the two-hit models versus in yours to get these different shapes?
Yes. So the two-hit model assumes basically a kx to the n sort of constitutive relation where the n is prescribed, right? So it's an integer value that could vary. And there's an underlying rate of tumor acquisition, or sorry, rather hit acquisition that's independent over time. And then you have one free parameter that basically scales that to fit it to the data. So that's a two parameter model for a fixed n hit, if you will. In our model, the number of parameters we have are actually we have more parameters, but the parameter that we use for T cell recognition, there are two. There's a diversity parameter and there's a turnover parameter. And both of those are basically imputed as direct functions of what the T cell diversity and T cell turnover rate are for normal individuals in the population. So there are ways to measure, you can calculate the number of unique T cell clones as a function of age. And then there's a way to estimate T cell turnover through TREX, T cell receptor excision circles, or something like this, where people have found this in the blood, and it's a very reliable measure of turnover. Those two parameters will basically dictate the probability of recognition along any age group. And so there's one scaling there. But our second parameter, I mean, if you will, the second parameter that's coupled to this is the baseline incidence rate of cancer initiation. Does that answer the question?
Yeah, so is your curve here, is this a fit or is this a prediction out of previous fits?
My curve is a fit where the fitting the one parameter that changed from our constitutive modeling in the previous slide is, on average, how often does a tumor initiating event happen, and it's assumed to be constant for all age groups. So we're using that as our free parameter to fit.
Pretty good then. Thanks.
Thank you. Thank you.
Cool. All right. Thank you, Jason.
Thank you.
Once. Yeah.